Good afternoon, dear students. Today, we shall discuss the biochemical aspects of starvation and obesity. We shall discuss the biochemical aspects of starvation and obesity. What is starvation? Starvation is a severe deficiency in caloric energy intake below the level needed to maintain an organism's life. An organism's life maintenance needs a specific number of calories. To attain that particular number of calories, you need to give some food to the organism. If that little amount, that critical amount, if it is not is able to be supplied to the body, then that condition is starvation. We have two states, fasting and starvation. What is fasting? The human being especially, or even certain animals, they get food only at specified intervals. Certain canines are the animals who live on non-vegetarian food. That means they may get a kill sometime, and then an animal may be available or not available, and hence when they will get the next food, they do not. But as far as the human being is concerned, they have a habit of taking food in regular intervals, maybe 3 times a day. And then once the night food is taken, and in the morning, the time span is up in between 8 to 40 minutes. So the gap between the two meals, the night meal and morning meal, is in between 8 to 14 hours. If that much time is there, 8 to 14 hours after taking the last meal, if food is taken, then that is a fasting state. So fasting state is a maximum of 14 hours and a minimum of 8 hours. In between this gap period, we can call it as fasting state and the fasting metabolism. If the food availability is crossing 14 hours, and going beyond that, then we shall call it as a starvation. No more, it is fasting. So deficiency in caloric energy intake, it is in the form, it is the quantitative way of definition. This is the time frame of definition is 8 to 14 hours, and beyond 14 hours is the starvation. And in the caloric terms, the deficiency in caloric energy intake. Fasting being if no food is ingested after absorptive period 8 to 14 hours. Consequence of fasting is plasma level of glucose, amino acid, triglycerides falls down in the circulation. And now we'll go for overview of fasting. Insulin secretion falls while glucagon is activated. In the slowly fasting state, the insulin secretion slowly coming down and the glucagon secretion is activated. The fuel stores and enzymatic changes during fasting we shall discuss. In the body, what may be the amount of fat in a moderately built and then nutritionally neutral individual, and about 15 kg of fat in a 70 kg individual will be the fat store, and which is amounting to 135,000 kilocalories. So this is the storage energy in the body. And in the form of protein, 6 kg can be released. Normally, the protein is present in the muscles, various types of muscular cells, muscle cells. And then this protein, a part can be liberated and can be given in the dynamic form to produce glucose. How can protein use glucose? Gluconeogenesis. Then the stored form of carbohydrate is glycogen. This is a maximum of 0.25 kg, and which is amounting to 800 to 1000 kilograms. This is the available energy if food is not given for certain time. So once this energy resource is completely gone, then there is going to be, the organism cannot survive, which means death. Death is going to occur. What are the changes, enzymatic changes in the fasting? The flow of intermediates through pathways of energy metabolism. What are the pathways of energy metabolism? Carbohydrate metabolism, lipid metabolism, and protein metabolism, mainly energy metabolisms. They are broken down and producing energy. And in these various paths are controlled by 4 mechanisms. This energy metabolism is controlled by 4 mechanisms. One is availability of substrates, that is the carbohydrate, lipid, or protein. And allosteric regulation of enzymes, covalent modification of enzymes, and induction, repression of enzyme synthesis. These 4 mechanisms control it. And then there are going to be changes in the fasting. And the metabolic changes observed in the fasting are generally opposite to those described for well-fed state. One side is well-fed state, another is a fasting state. And generally all those metabolic changes are opposite in direction, except at 3 levels. One is glycogen phosphorylase level, second one glycogen phosphorylase kinases level, and hormone sensitive lipase level. Hormone sensitive lipase level. There is a break. Sorry for that. interruption. The metabolic changes observed in fasting are generally opposite to those described for the well-fed state. Exceptions we said. Glycogen phosphorylase. What does it do? Glycogen phosphorylase. It breaks down the glycogen to produce glucose. Glycogen phosphorylase kinases. These are the cascade of events which will connect the hormonal systems to that of the glycogen phosphorylase. And then hormone-sensitive lipase of adipose tissue. Again, hormones which can influence the lipase which is being present in the adipose tissue, and this is hormone-sensitive lipases. These mechanisms are operative in the fasting state. Then, the stimulation and inhibition of glycogen degradation is under the control of mainly glucagon and epinephrine. Glucagon and epinephrine increases in the fasting state, and they mediate through the cyclic AMP mechanism, and then they stimulate various adenylyl cyclase and produce a cyclic AMP, and cyclic AMP in turn activates various protein kinases, and they influences the main two enzymes, glycogen synthesis, glycogen breakdown, that is glycogenolysis and glycogenesis. Both of them are controlled by two important enzymes. One is synthesis is by way of glycogen synthase, and glycogenolysis is mainly the enzyme is glycogen phosphorylase. Both of them, once this is activated, that will be shut down. So this mechanism is operated by the cyclic AMP through various protein kinases, and then their ability to phosphorylate a phosphate moiety will be added to the both enzymes. Once the phosphate moiety is added to the synthesizing enzyme, that is the glycogen synthesis, then that will become inert, or otherwise it is inactive. Now here we want the available glycogen has to be broken down and produces glucose because there is a fasting state. So the stored glycogen need to be broken down. So for this purpose, the enzyme is glycogen phosphorylase. So the phosphorylation, or otherwise a phosphate addition by these kinases, activated kinases, will be activating the glycogen phosphorylase. So now glycogen phosphorylase becomes activated, and the glycogen is broken down and the glucose is produced. And that is the theme behind the glucagon influencing the cyclic AMP, and in turn influencing the various kinases, and then kinases will be activating the glycogen phosphorylase, and glycogen is degraded in the fasting state. Similarly, epinephrine is also having the activity through the beta adrenergic receptor in the muscle and liver, and calcium is also having the same capability to activate the phosphorylase kinases. So there are three mechanisms. And then in the fasting state, what are all the various organs involved to combat the problem? The liver, adipose tissue, resting skeletal muscle, brain, and kidney. So 5 organs will participate in the fasting state to combat the problem of fasting metabolism. So liver, adipose tissue, resting skeletal muscle, brain, and kidney. What are the events that are going to happen here? We shall discuss. In the liver, in the fasting, what happened to the carbohydrate metabolism? Fasting, that means the energy, the circulating carbohydrates is reduced. That is the main energy source is glucose. So glucose is being utilized by the cells in the over period of fasting. Then what is needed? The glucose has to be replenished. How it can be replenished? By putting the stored carbohydrate. What is the stored carbohydrate of the body? Is the glycogen. So glycogenolysis. How can? Through the glucagon increase, through the epinephrine stress, both will produce activate the glycogenolysis main enzyme, glycogen phosphorylase. And second one, once one side this is going on, and then it cannot replenish. Just now we saw only 0.2 kgs only is the glycogen, the available glycogen. The remaining main energy sources are the fat and the proteins. Increased gluconeogenesis. What are the main sources for the gluconeogenesis? The used up carbohydrates like the lactate and the pyruvate. They are the source for neogenesis. What is the definition of neogenesis? Synthesis of glucose from non-carbohydrate sources. Non-carbohydrate sources. That is the main definition. So what are the non-carbohydrate sources? One is the lactate. Second one is the main one is the proteins. From the proteins, amino acids, various amino acids. Mainly the alanine. Alanine is the main source. And then the very small quantity can also be obtained from the glycerol part of triglycerides. When triglycerides are being utilized, then glycerol molecule will be coming out, and that can be a source. And in the odd chain fatty acid degradation, that is the oxidation, producing energy. Then the last piece of degradation is going to be propionic acid. This is a beta oxidation. Every 2 are being broken down, hydrolyzed, and then becoming acetyl-CoAs, and then they are being utilized for the energy sake. They have been metabolized for the energy sake. And the last one in the odd chain carbohydrates is going to be 3-carbon moiety, which is propionic acid. So that is a source for the neogenesis. So except that, the small part of glycerol, a small part of the odd chain carbohydrate, which is very, very, very small. So in stoichiometrically, the amount is very, very little, and hence in the neogenesis, the main material for the source of material for the synthesis of glucose in the neogenetic pathway, mainly lactate, pyruvate, and glucogenic, all the majority of the amino acids, glucogenic amino acids, and the last is glycerol and protein acid. In the fat metabolism, what are the changes in the liver? Increased fatty acid oxidation. If the fatty oxidation is being taken, becoming faster, becoming activated, then the speed will be increased. Again, same increased synthesis of ketone bodies. Ketone bodies we have held. And ketone bodies are important. They are synthesized from the acetyl-CoA, and then these ketone bodies will be the fuel in the emergency. When compared to the fatty acids to be broken down in certain tissues, ketone bodies oxidation is easier, and hence it is preferred by various certain organs. Increased gluconeogenesis. Gluconeogenesis is favored by activation of fructose 1,6-bisphosphate due to drop in its inhibitor. You know, the inhibitor is fructose 2,6-bisphosphate. This is inhibited in the fasting, and then there is going to be activation of neogenesis and induction of phosphoenol pyruvate carboxykinase by glucagon. Glucagon directly influences the enzyme PEP carboxykinase, phosphoenol pyruvate carboxykinase. One of the important enzyme in the gluconeogenesis is phosphoenol pyruvate carboxykinase, and this is going to be influenced by glucagon and activated, and then the neogenesis is increased. And here, regarding fat metabolism in the liver, acetyl-CoA does not act as a substrate for neogenesis. Just now I said it acts as it acts on pyruvate carboxylase only to stimulate it. And it is an acetyl-CoA is an inhibitor of pyruvate kinase, thus pushes pyruvate towards the gluconeogenesis in this fasting state. The major metabolic pathway in the liver during starvation. This is glycogen is going to be producing the glucose, and then the fat acids, acetyl-CoA, they are going to produce ketone bodies. So there is going to be gluconeogenesis, there is going to be ketogenesis as the fasting is proceeding. And then there is regarding the Carnitine shuttle and enzymes. What is this Carnitine shuttle? In the mitochondria, all the fatty acids need to be transported, at least all the long chain fatty acids need to be transported. And they are transported by a transport or a boat called Carnitine. and carnitine palmitoyl transferase 1 and carnitine palmitoyl transferase 2, two enzymes, the two enzymatic subparts available in the inner and outer mitochondria. And these fatty acyl-CoA is going to join carnitine, and then again, while crossing the so-called the barrier of the two membranes of mitochondria, entering into the inside of the mitochondria, the carnitine leaves the fatty acid, and again acetyl-CoA is being given back to the the fatty acid, and fatty acyl-CoA is going to be synthesized. And that is how the outer fatty acyl-CoA, it crosses the membrane barrier, and the inner acyl-CoA is being again re-synthesized. And for this purpose, carnitine goes a roundabout from inner membrane to outer membrane. And this is what the carnitine shuttle. Unless carnitine is available sufficiently, the fatty acid transport is not proper, and energy metabolism is going to be hampered. So carnitine shuttle activated. And then increased synthesis of ketone bodies, just now we said. Liver cannot use ketone bodies, but it produces more ketone bodies. It is an important source of fuel for, especially ketone bodies are the important fuel for skeletal muscle in the rest of the muscle, renal cortex. Brain normally prefers glucose, but when glucose is a little bit deficit, then ketone bodies are preferred. And the cardiac muscle in the less stressful state prefers fatty acids, but when cardiac muscle is undergoing some stress, then prefers ketone bodies and a bit of glucose. This is what the preference of the metabolic fuels. And adipose tissue in the fasting. Adipose tissue is the main storage form of the fat. And the number of adipose tissue cells are also depending on genetic constitution of the body. The adipose tissue size increases and decreases depending upon the energy excess available or energy deficit is there. Then basing on that, the size of the fat cells are going to be increased or decreased. And that is what the theme here, the fat tissue theme. The fat increased degradation of triglycerides. They are mainly hormone sensitive lipase, fats. Increased release of fatty acids, then once they are broken, fatty acids are released, glycerol and fatty acids are released, and they are circulating in the blood along with the albumin molecule, and then there is a raise in the fatty acids. Decreased uptake of fatty acids, lipoprotein lipase of adipose tissue is low, and hence the adipose tissue won't accept the fatty acids. That means fatty acid degradation is increased, fatty acid synthesis is reduced in the adipose tissue in the fasting state. What about in the skeletal muscle? Skeletal muscle, during the first 2 weeks, fatty acids from the adipose tissue coming to the towards ketone bodies from the liver used as a fuel to these skeletal muscles. Regarding the protein metabolism in the skeletal muscles, alanine and glutamine are the quantitatively the most important gluconeogenesis amino acids used as substrates in the. So gluconeogenesis precursors are released, and ketone bodies are utilized preferentially. Fatty acids are also being utilized at the in the muscles, in the muscle tissue. What about the brain? You see, brain in the well-fed state, all the glucose, the completely 100% glucose, the brain depends on completely glucose metabolism in the well-fed state. But once the glucose levels are reducing slowly in the blood levels, and then that means it senses fasting and slowly switches on to the alternative fuels. The brain switches on to the alternative fuels. And those are, one is amino acids, another is the ketone body, acetoacetate, and mostly hydroxybutyrate, another ketone bodies. Ketone bodies are 3: acetone, acetoacetic acid, and hydroxybutyric acid. Beta means 3 are beta hydroxybutyric acid. So this is going to be preferentially used in the fasting state by the brain with some amount of a must. Glucose, some amount of glucose is a must, and this glucose is being prepared in the body through gluconeogenesis. And kidney is another organ which mainly participates in the ketone in the starving fasting state. Kidney expresses the enzymes of gluconeogenesis, including glucose 6-phosphatase, important enzyme. In late fasting, about 50% of neogenesis occurs. Provides the neogenesis in the kidney tissue, and then especially the kidney also participates in the neogenesis process. Provides compensation for the acidosis that accompanies the increased production of ketone bodies. And this is how mainly the ammonia that is released is mainly stored as the glutamine using glutamic acid, or using alpha-ketoglutamate acid. Initially glutamate is being produced, and again further ammonia addition producing glutamine, and some amount of ammonia directly being excreted into the urine as ammonia, and so with the help of some hydrogen ion, the ammonia becoming ammonium, and ammonium is being excreted into the urine, and that's how some amount of ammonia is being thrown out through the urine, and some ammonia is buffered into the glutamine. So this is another adaptation. And if the starvation is continuous persistently for a long time, it is going to be the malnutrition, protein, calorie malnutrition in case of children, and this is carbohydrate emaciation in the elderly people, and these are the younger people, especially African population. Most of them are not having enough energy supplies through either carbohydrate or fats, and hence they suffer a long time starvation, and then their bodies are emaciated. And there are, in the children, there are 3 kinds of malnutrition, which you already know: the protein calorie malnutrition, kwashiorkor and marasmus, and in between the marasmic kwashiorkor are the spectrum of it. And so 3 phases can be identified. The malnutrition can be identified in the children. One is kwashiorkor, protein malnutrition, energy malnutrition, marasmus, both of them including protein, marasmic kwashiorkor, a mixed one, the spectrum one. And that is all are observed in the African children. Metabolic alterations of starvation, you see, there are 4 stages. First stage, increased glycogenolysis. Second stage, increased gluconeogenesis. The third stage, increased glycolysis and fatty acid oxidation increased. And in the 4th stage, increased ketogenesis. And now, as the ketone bodies are highly being circulating in the blood, some amount of ketones are seeped into the urine. And you know, a test called ketone body test in the urine, that is Erodhar's test, you will do it, and a purple colored ring you will see, and that is because of increased ketone body circulating in the blood to supply the energy needs of various organs. And then various hormones that are influencing are norepinephrine, epinephrine, and thyroid hormones. They are all going to be altered. Norepinephrine is secretion is reduced, epinephrine secretion is increased, and then in the adrenal gland, and then thyroid secretion is going to be decreased with the starvation. And the differentiation in well-fed and fasting states of the human body are, these are the various table form of how the preferences are being given in the various organs for various substrates in the well-fed state and in the fasting state. In the liver, in the well-fed state, glucose and fatty acids are utilized preferentially. In the fasting state, only fatty acids. In the resting skeletal muscle, glucose and fatty acids both will be utilized. But whereas in the fasting state, it goes for the fatty acids and ketone bodies. You see, at the level of, for the sake of, as glucose is getting reduced, the ketone bodies are preferred by the resting muscle. In the cardiac muscle, normally in the well-fed state, fatty acids are being utilized, whereas in the fasting state, fatty acids, amino acids, and ketone bodies, all three will be utilized. In the adipose tissue, the glucose is utilized, but then whereas in the fasting state, fatty acids are used. In the brain, mainly in the well-fed state, we have seen the bar diagram where glucose only the preferred, and whereas in the fasting, glucose and the later ketone bodies. RBCs at any stage, it is the only glucose. And hence, if the glucose is not available, then RBCs are going to suffer. They are unable to carry the oxygen properly. And well-fed state and fasting state, the sources of glucose, you see, the sources of glucose is from food, whereas in the fasting state, it is from the neogenesis and glycogenesis. The fate of glucose in the well-fed state is, it is glycolysis, and the formation of glycogen is main, and then still excess glucose is there, triacylglycerides will be synthesized. But in the fasting state, it is going to be glycolysis. And sources of fatty acids is in the well-fed state is from the food. But whereas in the fasting state, the storage triacylglycerols are going to be broken down, not synthesized. The fate of fatty acids in the well-fed state, beta oxidation, whereas in the fasting state, incomplete beta oxidation is ketogenesis. So more of ketogenesis. The well-fed state, all the time fatty acids are going to be beta oxidized. And then sources of amino acids in the well-fed state is from the food. But whereas the amino acid source is for the sake of breaking down of, for the sake of producing glucose from breaking down of muscle proteins. And amino acids fed in the well-fed state is protein synthesis, whereas in the fasting state, they are going to produce glucose, that is neogenesis. So hormones, in the well-fed stage, insulin is going to be increased, whereas in the fasting state, glucagon is increased, adrenal is increased, cortisol is increased. The response of the body is, in the well-fed state, is hyperglycemia, glycogenesis, and protein synthesis. All are constructive things happens in that. Constructive metabolism are going to happen in the well-fed, whereas in the fasting is the catabolic, that is the hypoglycemia, glucose is utilized, glycolysis, ketogenesis, and proteolysis. So these are the various changes that are occurring in the fasting state. Now obesity, on the contrast, there is a deposition of fat in the body in excess. So that is important. Deposition of fat in the body in excess, mainly in the adipose tissues. It is a disorder of body weight regulatory systems. It is a disorder of body weight regulatory systems characterized by accumulation of excess body fat. Currently, obesity is an epidemic because abundance of food is available and physical activity is reduced in the human population. That's why it is going in an epidemic form. You can see an example how children are eating food without need. So various fatty foods consumption, especially in the advanced world. Obesity and health. What are the implications of this fat? This fat is a healthy sign up to only as far as the reasonable fat accumulation is healthy, and beyond that it is a disease. Obesity is also a disease. What is that disease? What is the disease and what is the effect of this obesity on other diseases? In other diseases, chronic diseases. Risk factor. This is a risk factor. If obesity is there, the other things are more likely to get your body likely to get the other disorders or diseases. What are they? Type 2 diabetes mellitus. Once obesity is there, there is going to be what is called insulin resistance. And then once resistance is there, its effect on the glucose metabolism is reduced. There is going to be build-up of glucose levels. And that is what is that type 2 diabetes mellitus. Insulin insensitive diabetes mellitus is called as type 2 diabetes mellitus. That is here, insulin even though present, the capacity to work is reduced. Its sensitivity is reduced. and hence there is diabetes mellitus. Hypercholesterolemia, cholesterol levels crosses the healthy limits. That is, now we call it as the preferable or otherwise desirable cholesterol, not normal cholesterol, desirable. That is 200 milligrams per deciliter. High plasma level of triacylglycerols, fatty acids levels are going to be increased. Hypertension, there is going to be increased blood pressure. Coronary heart disease is going to happen, likely to get, and some cancers are related to obesity, and gallstones, stones formed in the gallbladder. Arthritis, there is going to be inflammation in the joints, and there is gout. So all these ones are related to the obesity. They are associated with obesity. Assessment of how do we assess the obesity? We take the weight and try to incorporate in the health charts. The thousands of people body weights are being taken, and healthy lean people's weight is taken, and then maps are being constructed, and we'll try to incorporate the individual's body weight, how much it is, and the both height and weights are coming into the effect here, and a particular certain height and a particular weight, and then whether they are falling in the desirable level or beyond the desirable levels. And the next one is overweight, and the last one is the obese, and the health surveys have constructed each and every country's maps. These are the healthy weight maps. And basing on this, obesity can be pronounced. And the second way, indirect way of assessing without having these charts and the national surveys, that is BMI, Body Mass Index, with the two important parameters. One is height and another is weight in kgs, or the height in inches and the weight in lbs. So, but the preferable one is in meters height and then kgs weight. And basing on that, the BMI is weight in kgs divided by height in meter square, or else in the British system, weight in lbs divided by height in inches square. So anatomical differences in fatty deposition is another way of pronouncing obesity. Biochemical differences in fat deposits can be obtained, and there can be obesity recognized. The number of fatty cells can be also giving indirect assessment of obesity. Anatomic differences in fat deposition. You see, the males have a specific shape. This is what is called as the abdomen. Is the fat is mainly deposited in the abdomen of the males, whereas the females, you see, their fat is deposited mostly in the waists. So this fat in the abdomen, male type of obesity, is similar to that of an apple, and hence this is called as apple-shaped obesity. Whereas in the females, you see, it is mostly in the hips and the legs, rather than the thighs, hips and thighs, and hence this shape is mimicking a pear fruit. So apple or pear. So basing on that, males are apple-shaped upper body obesity, whereas females are having a pear-shaped lower body obesity. So these are otherwise a different types of anatomical distribution of fat in genders, male and female, in a little different way. Apart from this, a lot of fat can be accumulated in the omentum of both the people. You see, the weight by height square. Basing on that, this body mass index, now the optimal BMIs are classified. 18.5 to 24.9 is a normal BMI. Less than this is underweight. And more than this is overweight. Up to what level? 25 to 29.9. If the BMI is crossing 30 and in between 34.5, 34.9, it is called as class 1 obesity. And 35 to 39.9, class 2 obesity. And more than 40 is class 3 obesity. You see, diagrammatic representations are being given. And more than 40, again, is called as severe obesity. And 40 to 49.9, this is morbid, diseased obesity. And over 50 is super obesity. You may be seeing the Japan people, the Razalas, sumos. And then nearly 2/3 of American adults are overweight and more than 1/3 are obese. So this is about American scenario. And India, obese people are not that much, but overweight is increasing. Biochemical differences, what are they in the regional fat depots? Abdominal fat cells are much larger and have rate of fat turnover, lower body fat cells. Abdominal adipocytes are hormonally more responsive. Men tend to accumulate more mobilizable fat. Substances released from abdominal fat are absorbed via portal vein and have direct access to liver. And this fat is having more influence on insulin resistance. The number of fat cells, this is more important. The number of fat cells usually are reasonably fixed in individuals. And the fat cells size is going to be increased or decreased. And once the obesity starts, once the remnant, or rather the adipose tissues in the quiescent stage starts activated and becomes active fat cells, then they will not go back. And then these are pre-adipocytes. The pre-adipocytes once becomes adipocytes, they are going to harm, they are going to shelter the fat, and then once fat cells are increased, they are not reduced normally. Only in the exceptional state they can be. And most obesity start to involve an increase in both the number and the size of adipocytes. Fat cells once gained are never lost. In the new experiments, there is an evidence that they can be with great difficulty. The observation that fat cells are never lost emphasizes the importance of preventing obesity in the first place. Body weight regulation, genetic contribution to obesity. Identical twins have very similar BMI. If both parents are obese, child have 70 to 80% of chance, you see. If parents are lean, then child has just only 9%. So genetic predisposition is there. Environmental and behavioral contributions. What is that? Japanese in the Japan have an average BMI of 20, but once they are migrated to USA and they are getting the habits of the Americans, then the average BMI is 24. And molecules that influence obesity. What are the molecules that are influence the obesity? The central point of the what you call the food. This is thalamus. Thalamus is controlling the food intake. Afferent satiety, mainly the satiety center is there in the hypothalamus. There are afferents from various places come to the satiety and influences, and efferents will go, signals will go. What will happen? Insulin from the pancreas, and a hormone called ghrelin from the stomach, and from the leptin, from the adipose tissues, a hormone called leptin. So important hormones of obesity, leptin and ghrelin, and with the help of insulin, and from the intestines there are two more hormones, CCK, cholecystokinin, and PYY, neuropeptide Y. Y means tyrosine. A tyrosine is more present in this peptide, and hence it is given neuropeptide Y. So intestine produces PYY, and then the CCK, it's cholecystokinin. So please remember, obesity is being influenced by various hormones: insulin, leptin, ghrelin, cholecystokinin, and neuropeptide Y. And these are the signals given into the hypothalamus, and the thalamus gives signals in case of undernourished, increased appetite to produce more appetite, to decrease the energy expenditure. And that is the action that will be taken by the hypothalamus. In case of overnourished, then these hormones are cut short, and then there is the signal is different signal is decreased appetite, so not to take food, and then increased energy expenditure, more energy to be expended. Other factors such as availability of palatable, energy-dense foods, poor food choices, and then mediated by complex neuronal pathways. Metabolic changes observed in obesity. Predominant effects of obesity include the dyslipidemia, glucose intolerance, insulin resistance, expressed primarily in the liver, muscle, and adipose tissues. Metabolic syndrome called syndrome X is associated with the obesity. Dyslipidemia is due to insulin resistant, hormone sensitive lipase, circulating fatty acids, and carried to liver, converted to cholesterol and VLDL. That's how dyslipidemia occurs. The dyslipidemia is also due to the metabolic effects of obesity. Dyslipidemia, insulin resistance in adipose tissues is go on influencing the liver, increased free fatty acids. The liver is producing more of cholesterol and triglycerides. Excess cholesterol triglycerides are released as VLDL, and increased blood triglyceride hypercholesterolemia. Okay, increased risk of coronary heart disease. Then molecules that influence obesity are neuronal signals, hormones circulating in the blood. Hypothalamus releases different signals, influences appetite and energy expenditure. And circulating hormones, as I said just now, leptin, resistin, adiponectin. Leptin, resistin, adiponectin. They are the afferents to hypothalamus. And efferents from hypothalamus are nerves, CNS, norepinephrine, dopamine, and serotonin. These are the main control is from the hypothalamus. And hormones of adipose tissue, they are the circulating hormones, insulin and CCK. Insulin is from the pancreas, and then CCK is coming from the intestines, and then ghrelin is from the stomach, and adipose tissues producing leptin and resistin and adiponectin from the adipose tissues, and nerves and CNS through producing norepinephrine, and then dopamine and serotonin. They are all able to, a concerted mechanism controls the adipose tissue fat results. Leptin is the hormone product of the obese gene, secreted by fat cells, produced proportionally to adipose mass. Acts on the hypothalamus of the brain to regulate the amount of body fat through the control of appetite and energy expenditure. Leptin secretion suppressed by starvation, enhanced by well-fed state. Suppressed by starvation, enhanced by well-fed state. Ghrelin, peptide A, secreted primarily in the stomach, the only known appetite-stimulating hormone in humans. Injection of ghrelin in the rodents increases food intake, decreases energy expenditure. Decreased energy expenditure decreases fat metabolism. And thank you. This is a brief on the starvation and obesity. So mind you that everybody will maintain health through ideal body weight. Thank you very much.